Bacteriocins are antibacterial proteins produced by bacteria that kill or inhibit the growth of other bacteria. Being natural products, bacteriocins are more accepted by consumers as natural food preservatives compared to chemical preservatives. Pediococcus acidulacti KP10 is isolated from a traditionally prepared milk byproduct was previously found to secrete bacteriocin with inhibitory activity against Listeria monocytogenes, the deadly causative agent for listeriosis, and also susceptible to vancomycin, an inhibitor of cell wall synthesis which is broadly effective against multidrug-resistant pathogens. This video highlighted the steps developed for large-scale prototype production of bacteriocin by Pediococcus acidulacti KP10 based on 100-liter stirred tank bioreactor, which has been maximized to address both biologic and economic concerns. The stock culture of Pediococcus acidulacti KP10 maintained in freeze-dried form is treated on agar petri dish plate. The single colony from the petri dish is inoculated into flask containing liquid growth medium and incubated in an incubator shaker for 18 hours. This culture is used as a standard inoculum. Cultivation in the seed bioreactor with a volume of 10 liter is initiated by the inoculation of 1 liter of a standard inoculum. This means that the size of inoculum used is 10% volume per volume. The cultivation is carried out for 18 hours with optimal control of dissolved oxygen tension and the culture pH until the culture reaches the late exponential growth phase. To start the fermentation for the production of bacteriocin, the culture from the seed bioreactor is transferred aseptically into the sterilized 100-liter stirred tank bioreactor containing optimal medium formulation. During the fermentation, the dissolved oxygen tension is controlled at optimal value higher than 15% saturation by the manipulation of agitation speed and airflow rate. The fermentation is carried out for 24 hours. Upon completion of the fermentation, cell is separated from the fermentation broth using continuous centrifuge. Primary extraction of bacteriocin from the culture supernatant is performed using tangential flow filtration method. The concentrated filtrate containing bacteriocin is then purified using aqueous two-phase system, employing polyethylene glycol and citrate buffer as the phases.
Put this there.
The process is carried out in 5-liter mixing tank with optimal agitation and separation time. The high purity bacteriocin is captured in the polyethylene glycol as analyzed by the SDA speech. The bacteriocin in polyethylene glycol is formulated by freeze-drying method using skimmed milk and glycerol as the protective agent and bulking materials. The bacteriocin activity is analyzed again by food pathogens such as Listeria. The commercial preparation of bacteriocin in freeze-dried form is stable for a long storage period. We would like to thank the Higher Education Department, Ministry of Education, for funding this prototype research grant scheme PRGS project Biomanufacturing of Bacteriocin by Pediococcus acidulacti KP10.